name is Yaswanth Reddy. I am studying at University Institute of Technology in the branch of ADS. I did a project on cancer detecting by using the brain tissue MRI scan under the guidance of SIC AIML branch. Here you can see first I download the dataset from the Kaggle, which is the dataset is very huge. You can see how I downloaded by use since the data is stored in H5 Python file. So I use it to extract the data like that only. Here you can see this one importing H5py. Then if we go like this inside this is the data how it is collected and the path I had shown. Here you can see the path where I downloaded to VS Code. Next if we go little bit further, now I'm seeing the shape of the training data, which is training. There are two sets. One is the training label and training images. You have to combine the two to know which is the which tissue is like cancer, which is not. So here you can see the image shape of the image shape and the label shape are similar, which is like 2,62,244. Like that. Here I have taken only 1 lakh dataset because it is the huge. So I taken from every two classes like class 0 and class 1. Class 0 means with cancer, class 0 means without, class 1 means without cancer. So 1 lakh I taken, totally 2 lakh, which I reduced nearly 31%. See, you can see the class 0 is 1,31,000 and class 1 is 1,31,000. Here I used to take that one. Next, if you go little bit further, this is normally image loading. And here I use it, this is the code for the random saturation, because when I use the normal data, since the size of the cells were different, it is difficult to train the CNN model based on the size. So I use the saturation. You can see here the printing of class 0. First column is for first row is for the class 0, which is like without cancer, and second one is for with cancer. You can see the class 1. The images numbers also I shown 1, 2, 3, 4 like that. And here is the thing, which is like before saturation and after saturation, the image how it is. Similarly, I used it to generate everything like that only. Then here you can see I used the training batch size as 124 and the validation batch size as 64, because the focusing on the less images better for the validation compared to that one. And this is my own model which I create by my own self by taking the reference of VG16 model. In VG16 model, you have everywhere you use only max polling, but here I used average polling also at 2 times. Next, when you compare this model with the VGG model, the parameters were nearly 55% decreased, and the MB also, this is for nearly somewhere like 150 MB. When you take VGG, it is 500 MB nearly. So there is a huge difference. And then this is for my approach and training. Here is I use it for the callback, early stop, and some functions. Here you can focus on the valid accuracy. If the valid accuracy is decreased more than 2 to 3 times, it check the patient's value how much we given. Here I given nearly 2. So if 3. If the valid accuracy is decreased more than 3 times, then the function will stops. So approaches will stops. So here you can see. For my last approach number 5, the difference between the accuracy and the valid accuracy is less than 7%. So I consider this as the normal. So I taken this one. Since you can see my model next is it is for the testing. The accuracy is like 86.96 for test accuracy, and everything is normal. Like you can see the confusion matrix. And last one is ROC graph, which is like 0.95 I got. Thanks.